Chapter number 4. Tools of Genetic Engineering 2. Cutting and Joining of DNA. Genetic engineering is used for the production of valuable polypeptides, insulin, interferon, growth hormones, and in the transfer of NIF genes, control of genetic diseases, etc. There are three basic steps of genetic engineering: 1. Isolation of DNA fragments from a donor organism, 2. Insertion of an isolated donor DNA fragment into a vector genome, and 3. Growth of a recombinant vector in an appropriate host. There are various biological tools which are used to carry out manipulation of genetic material in cells as well. For example, enzyme, foreign or passenger DNA, vector or vehicle DNA, gene library. This chapter deals with enzymes used for cutting and joining of DNA molecules. Enzymes used in genetic engineering. There are many enzymes which are used in genetic engineering as an important biological tool. However, the important enzymes required for genetic engineering are restriction endonucleases, DNA ligases, exonucleases, DNA polymerases, alkaline phosphatase, polynucleotide kinase, terminal deoxynucleotide transferase, reverse transcriptase, etc. Restriction endonucleases are the special types of endonucleases that cut DNA at specific sites. Recombinant DNA technology is based totally on different types of enzymes. Some of the enzymes used in gene cloning are given in Table 4. and described in this chapter. See the Table 4.1, Enzymes used in DNA cloning. 1. Exonucleases. These enzymes act upon genome and digest the base pairs on 5' to 3' ends of a single-stranded DNA or at single-strand nicks or gaps in double-stranded DNA, Figure 4. On the basis of mode of action, the exonucleases are grouped into different types. See the figure. Figure 4.1. Action of exonuclease A and endonuclease B. Lambda exonuclease. This enzyme removes nucleotides from 5' end of double-stranded DNA. Consequently, an improved substrate for terminal transferase is formed. Therefore, this enzyme is used for 5' end modification. The mode of action of this enzyme is given below. See the figure. Exonuclease type 3. This enzyme removes nucleotides from 3' end of the double-stranded DNA fragment. Consequently, an improved substrate is formed which is used in gene manipulation. This enzyme is used for 3' end modification of DNA fragment as below. See the figure. 2. Endonucleases. They act upon genetic material and cleave the double-stranded DNA at any point except the ends, but their action involves only one strand of the duplex. Figure 4.1b. Figure 4.2. Post-controlled restriction and modification of phage I. In E. coli strain K and strain B. 3. Restriction endonucleases. The restriction enzymes are called as molecular scissors. These act as foundation of recombinant DNA technology. These enzymes are present in bacteria and provide a type of defense mechanism called the restriction modification system. Molecular basis of these systems was elucidated first by Werner Aber in 1965. However, there was no understanding about cutting and joining of DNA molecules using DNA enzymes before 1970. During the early 1950s, it was discovered when phage infects E. coli, phage DNA is delivered inside the cell. Post-control restriction modification system provides defense to E. coli. Bacterium protects itself through 1. Restriction mechanism, i.e. identifies the introduced foreign DNA and cuts into pieces called restriction endonucleases, and 2. Modification system, example, methylation of certain bases of its own DNA by methylase so that phage lambda X expressed restriction endonuclease could not cleave its DNA. Figure 4.2 shows host's restriction modification system. If phage lambda propagated on E. coli strain Ka is allowed to infect E. coli strain B, the efficiency of infection is very low B. The progeny phage obtained from plaques of B strain can reinfect E. coli strain B with high efficiency C. But infection efficiency of these progeny phage on strain K is also very low D. There are 3 gene loci HSDS, HSDM and HSDR that control this system. The HSAS encodes a protein which governs the specificity of these system. The HSDM gene product acts as modification enzyme methylase which interacts with HSDR gene product restriction enzyme in the cleavage process. It may be explained that progeny phage particles first grown on strain K and then strain B are restricted when grown on B strain. But the progeny phage particles from strain B are not restricted when reinfect strain B again, they are restricted when grown on strain K. This shows that the efficiency with which phage grows on a specific host depends on the host strain on which it was propagated previously. It was confirmed when the restricted phage is adsorbed on the surface of restrictive host and injects its DNA, it is broken down quickly by restriction endonuclease. Hence, the restrictive host protects its own DNA from degradation and modifies its DNA enzymatically. Thus, the restriction activity is provided by two enzymes, nuclease and methylase, the genes of which reside on the host chromosome or plasmid. There are three different types of restriction endonucleases: type 1, type 11, and type IL. Each enzyme differs slightly by different mode of action. Type 1 and type 11 are large multi-subunit complex which contains both endonuclease and methylase activity. The different bacteria consist of different restriction endonucleases, and in correspondence to these the methylase 2 are produced. These enzymes occur naturally in bacteria as a chemical weapon against the invading viruses and cut both strands of DNA when certain foreign nucleotides are introduced in the cell. These enzymes cleave a DNA to generate a Nic with a 5' phosphoryl and 3' hydroxyl termini. A. Type 1 restriction enzyme. The BK restriction and modification enzyme systems of E. coli have been termed as type 1 endonucleases such as ECOB and ECOC. They are complex nucleases which function simultaneously as an endonuclease and a methylase. They move along the DNA in a reaction and require Mg+4, S-adenosine methionine and ATP as cofactor. They are single multifunctional enzymes consisting of three different subunits: 1. Restrictive subunit, 2. Modification subunit, and 3. Specificity subunit. The third subunit specifies the recognition sites. The enzymes show specificity for recognition site but not for cleavage site, i.e., they recognize specific sites within the DNA but do not cleave at these sites. This results in production of the heterogeneous population of DNA fragments. Type 1 enzyme recognizes 15 BP long and cleavage site is about 1000 BP away from the 5' end of the recognition sequence TCA, which is present within 15 BP long recognition site. The adenine position in the restriction site may be methylated. Such DNA molecules are resistant to type 1 endonuclease whose recognition sequence is methylated at a residues on both the strands. However, when the recognition sequence is unmethylated, only then DNA is cleaved as below. Eco K5 double AACNNNGTCGC -3 3. TTTG none CAGCG -5 Here shows methylation at adenine position. N is unspecified bases and N is complementary bases to N. The unmodified DNA molecules as restricted, i.e., cleaved by restriction endonucleases. B. Type 2 restriction enzymes. Type 2 restriction enzymes were first isolated by Hamilton Smith during 1970s. They are simple, consisting of single polypeptide chain and require no ATP for degradation of DNA. They have separate activities for cleaving endonuclease and methylation methylase of DNA molecules. They are most stable enzymes that require Mg+ as cofactor. Daniel Nathans first used these enzymes for mapping and analyzing genes and genomes. The first one type 2 restriction enzyme isolated and characterized was from Escherichia coli strain Re. Hence, it was named as ECO1. For the discovery of restriction enzymes, W. Arbour, H. Smith and D. Nathans were awarded with Nobel Prize in 1978. See Table 4.2 List of suppliers of restriction enzymes. Thousands of restrictions enzymes have been discovered and some of them are commercially available. Table 4.2. Out of above 3 types of restriction endonucleases, type 11 are used in gene manipulation. Type 1 enzymes recognize a specific sequence of DNA molecule but cut elsewhere, and type 2 make cuts only within the restriction sites and produce 2 single-strand breaks, 1 break in each strand. Type 2 enzymes are the most important ones. As a result of their action, the broken nucleotides form a DNA duplex which exhibit 2-fold symmetry around a given point. In some cases, cleavage in 2 strands are staggered to produce single-stranded short projections opposite to each other with blunt or mutually cohesive sticky ends which are identical and complementary to each other. These complementary sequences are also known as palindrome sequences or palindromes. Therefore, when read from both strands have the same sequence. They are specific in recognizing the nucleotide base pairs on both the strands and cut exactly within the same recognition site. Recognition sites are the palindromes or palindromic sequences where most of type 2 restriction enzymes bind and cut a DNA molecule. Palindromes are the nucleotide pair sequences that the same when read forward, left to right, or backward, right to left, from a central axis. Of example, two strands are identical when both are read in the same polarity, i.e. in direction. For example, the following phrase is read same in either of the directions, left to right and right to left. The length of recognition sites of different enzymes varies, which can be 4, 5, 6, 8 or more nucleotide pairs. Table 4.3. There are many enzymes that recognize a hexanucleotide, a sequence of 6 nucleotides, whereas the others recognize tetra, penta or octanucleotide sequences present on DNA molecule. For example, EcoR1 restriction enzyme isolated from E. coli cleaves DNA at the hexanucleotide 5' GAATTC3, whereas EcoR2 isolated from the same bacterium recognizes PO4 end of pentanucleotide 5' CCTGG3. Recognition sequences of many type enzymes are palindromic sequences, i.e. they exhibit rotational symmetry. The nucleotide sequences are the same when read from 5 plus 3' in both the strands. For example, nucleotides of recognition sequences of EcoR2 are the same on both the strands as below. See Table 4.3, Source of Type 2 Restriction Enzymes, Cleavage Sites and Productions of Cleavage. Arrows indicate the recognition sites. Overhangs have 4 nucleotides. These ends are called sticky ends or cohesive ends. Because nucleotide bases of this region can pair and stick the DNA fragments again as given below, see the figure. On the other hand, some other type 2 restriction enzymes cleave both strands of DNA at the same base pairs but in the center of recognition sequence, and results in DNA fragments with blunt ends or flush ends. For example, He3 isolated from Haemophilus aegypticus, the order of enzyme 3,4 nucleotide long palindromic sequence and cuts symmetrically the both DNA strands and forms blunt ends. See the figure. There are some restriction enzymes whose recognition sites differ. Even then they produce identical sticky ends. For example, recognition sequence of BG2 is 5' AGATCT3, that of BAMH1 is 5' GGATCC3 and SAW is 5 GATC3. But all the three enzymes cut recognition sequences and produce 5 GATC3 sticky ends. Since fragments generated by each enzyme have identical sticky ends, they can join complementary sticky ends of one another. Table 4.3. The properties of type 2 restriction enzymes vary with its specificity, i.e. its changes with changing the reaction conditions such as lowering sodium chloride concentration, increasing temperature, replacing magnesium with manganese. For example, the recognition and cleavage sites of EcoR1 is normally a hexanucleotide sequence 5' GAATTC3, but it become a tetranucleotide sequence 5' AATT3 with the changes in reaction conditions. Moreover, the size of DNA, its base composition and the GC content of recognition sequences govern the number of cleavage sites of type 2 enzymes in a DNA molecule. For example, if all the 4 bases are present in a DNA molecule with 50% AT and 50% GC base pairs, the probability for a particular tetranucleotide is x14. The tetranucleotide shall be cleaved at a distance of every 256 base pairs. Nowadays, a large number of restriction enzymes are available commercially. Table 4.2. Some of the commonly used restriction endonucleases are given in Table 4.3. In 1983, R.J. Roberts has given an extensive list of restriction enzymes and the sequences recognized by them. Most of the enzymes share a common central nucleotide in their recognition sequence, for example, BAMH1, BG2, SAW3A. But these enzymes recognize different sites in DNA and produce identical single-stranded 5' tails which allow the joining of fragments generated by different enzymes within this set. The identical nature of the termini of cleaved DNA fragments from any organism is the very property which permits the annealing and subsequent ligation of the DNA from diverse sources. A. Nomenclature of restriction enzymes. The restriction enzymes are named based on some of the following principles. 1. Name of the organism is identified by the first letter of genus name and the first 2 letters of the species name to form a 3-letter abbreviation in the italic, for example, E. coli eco and H influenza equals HIN, etc. 2. A strain or type identified is written as subscript e.g. eco K for E. coli strain K, HIN for H influenza strain Rhode. 3. In such cases where the restriction and modification systems are genetically specified by a virus or plasmid, the extra chromosomal element is identified by a subscript, e.g., ECO-R1, ECO-P1, etc. 4. When a strain has several restriction and modification systems, these are identified by Roman numerals, for example Hind1, Hind2, Hind3 for H influenza strain Rhode, etc. These Roman numerals should not be confused with those in the classification of restriction enzymes into type 1. B. Type 3 restriction enzymes. The type 3 enzyme is made up of two subunits. One specifies TOR site recognition and modification and the other for cleavage and a reaction. It moves along the DNA and requires ATP as source of energy and Mg++ as cofactor. ATPase activity is lacking in these enzymes. Some examples of type 3 enzyme are HPA1, MABO2, FOC1, etc. They have symmetrical recognition sites and cleave DNA at specific nonpalindromic sequences. For cleaving double-stranded DNA, two sites in opposite orientation must be present. One strand of double-stranded DNA is cleaved about 25 to 27 BP away from the recognition site, which is located in its immediate vicinity. Recognition sequence of some of type 3 enzymes are given below. See figure. Since the cleavage products of these enzymes are homogeneous population of DNA fragments, they cannot be used for genetic engineering experiments. See oilier restriction enzymes. There are certain other restriction enzymes which do not fall under the type 1 to 3. Hence there is need for reclassifying restriction enzymes under the other categories from type 1 to type 4. Examples of the other restriction enzymes are ECO 571, MCR modified cytosine reaction, etc. ECO 571 is made up of single polypeptide. It shows both nuclease modification and nuclease activity. The S-adenosyl methionine stimulates nuclease activity of this enzyme. It bears some other properties that resemble with type 11 enzymes. Therefore, ECO 571 should be put under type 2 to type 4. The DNA of some bacteria and eukaryotes was restricted due to the presence of methylcytosine in the DNA. This was called modified cytosine reaction. In E. coli strain K, there are two types of MCR system, MCRA and Mcrbc. MCRA is encoded by a prophage-like element, whereas Mcrbc is encoded by two genes, Mcrb and Mcrc. Both the genes are present adjacent to the genes that encode type 1 endonucleases in E. coli strain K. Both the strands of DNA are cleave A, multiple sites between the methylcytosine, which is accomplished by GTP. The Mcrbc system is analogous to the RGB restricts glucoseless phage system, which is found in T1 coliphages. T1 bacteriophages consist of glucosylated 5-hydroxymethylcytosine in their DNA in place of cytosine. The phage DNA is restricted if glucosylation of 5-hydroxymethylcytosine is inhibited due to mutation of phage or host genes. Hence the host causes the restriction of phage. 4. S1 nuclease. It degrades the single-stranded DNA or single-strand protrusion of double-stranded DNA with cohesive ends. As a result of action of SL nuclease, cohesive ends are converted into blunt ends. Therefore, SL nuclease is used to remove the incompatible ends so that overlapping ends may be developed for annealing of two fragments of DNA molecules. The mode of action of SL nuclease is given below. See the figure. BAL31 nuclease. This enzyme is very specific single-stranded endodeoxyribonuclease. It degrades simultaneously both the 3' and 5' strands of DNA molecule. Consequently, shortened DNA fragments are formed. These fragments possess blunt ends at both the termini. Its mechanism of action is as below. See figure 5. DNA ligases. J. A. Mertz and R. W. Davis in 1972 for the first time demonstrated that cohesive termini of cleaved DNA molecules could be covalently sealed with E. coli DNA ligase and were able to produce recombinant DNA molecules. Figure 4.3. DNA ligase seals single-strand nicks in DNA which has O hydroxyl termini. There are two enzymes which are extensively used for covalently joining restriction fragments, the ligase from E. coli and that encoded by T4 phage. The main source of DNA ligase is T4 phage, hence, the enzyme is known as T4 DNA ligase. For the joining reactions, the E. coli DNA ligase uses nicotinamide adenine denucleotide NAD+ as a cofactor, while T4 DNA ligase requires ATP for the same. Both the enzymes contain a NH2 group on lysine residue. In both cases, cofactor breaks into AMP adenosine AMP monophosphate, figure 4.3, which in turn adenylate the enzyme A to form enzyme complex EAC. EAC binds to Nic containing 3' O and 5' P04 ends on a double-stranded DNA molecule. The 5' phosphoryl terminus of the Nic is adenylated by the EAC with 3' O terminus, resulting in formation of phosphodiester and liberation of AMP. Lehman, 1974. After formation of phosphodiester, Nic is sealed, figure 4.3. T4 enzyme has the ability to join the blunt ends of DNA fragments, whereas E. coli DNA ligase joins the cohesive ends produced by restriction enzymes. Additional advantage with T4 enzymes is that it can quickly join and produce the full base pairs, but it would be difficult to retrieve the inserted DNA from vector. However, cohesive end ligation proceeds about 100 times faster than the blunt end ligation. See the figure. Figure 4.3. Mechanism of DNA ligase enzyme AMP complex formation and sealing of Nic on a double-stranded DNA molecule. A. E. coli DNA ligase. B. T4 DNA ligase. A. Adenine. P. POG-R. Ribose. N. Nicotinamide. NMN. Nicotinamide mononucleotide. 6. Alkaline phosphatase. For joining a foreign DNA fragment when plasmid vector is treated with restriction enzyme, the major difficulty arises at the same time. Because the cohesive ends of broken plasmids, instead of joining with foreign DNA, join the cohesive end of the same DNA molecules and get recircularized. To overcome this problem, the restricted plasmid, i.e. plasmid treated with restriction enzymes, is treated with an enzyme, alkaline phosphatase, that digests the terminal 5' phosphoryl group, figure 4.4. The restriction fragments of the foreign DNA to be cloned are not treated with alkaline phosphatase. Therefore, the 5' end of foreign DNA fragment can covalently join to 3' end of the plasmid. The hybrid or recombinant DNA obtained has a Nic with 3' and hydroxy ends. Ligase will only join 3' and 5' ends of recombinant DNA together if the 5' end is phosphorylated. Thus, alkaline phosphatase and ligase prevent recircularization of the vector and increase the frequency of production of recombinant DNA molecules. The Nics between two 3' ends of DNA fragment and vector DNA are repaired inside the bacterial host cells during the transformation. The mechanism of action of alkaline phosphatase is as below, see the figure. 7. Reverse transcriptase. Reverse transcriptase is used to synthesize the copy DNA or complementary DNA, cDNA, by using mRNA as a template. Reverse transcriptase is very useful in the synthesis of cDNA and construction of cDNA clone bank. Until recently, it was known that the genetic information of DNA passed to protein through mRNA. During 1960s, Temin and co-workers postulated that in certain cancer-causing animal viruses which contain RNA as genetic material, transcription of cancerous genes on RNA into DNA takes place as most probably by DNA polymerase directed by virus RNA. Then DNA is used as template for synthesis of many copies of viral RNA in a cell. In 1970, S. Mizutani, H. M. Temin and D. Baltimore discovered that information can also pass back from RNA to DNA. They found that retroviruses possessing RNA contain RNA-dependent DNA polymerase, which is also called as reverse transcriptase. This produces single-stranded DNA, which in turn functions as template for complementary long chain of DNA. 7. DNA polymerase. This enzyme polymerizes the DNA synthesis on DNA template or cDNA template and also catalysis and exonucleolytic degradation of DNA. The DNA polymerase, investigated by Kornberg and co-workers in E. coli in 1956, is now known as DNA polymerase 1, DNA Pol 1. The other two enzymes are DNA polymerase 2, DNA Pol 2 and DNA polymerase 3, DNA Pol 3. These have almost similar catalytic activity. DNA Pol 1, molecular weight 109,000, has a single polypeptide chain of about 1.000 amino acid residues. The addition of mononucleotide to the free O end of a DNA chain is catalyzed by this enzyme. Also, it catalyses the other two reaction, i.e. 3 to 5' exonuclease activity, hydrolysing single nucleotide residues from 5' terminus, and 5 to 3' exonuclease activity, hydrolysing single nucleotide residues from 5' terminus. Function of DNA Pol 2 moles Wt. 120,000 is little understood. However, it catalyses 5 to 3 exonuclease activity. DNA pol 3 moles Wt about 140,000 is about several times more active than the other two. It is a dimer of DNA pol 3. It requires an auxiliary protein DNA copolymerase 3 and after combination, yields a DNA pol 3 copal 3 complex. Where there is preformed DNA template, it produces a parallel strand in the presence of ATP. DNA POI1 is susceptible to protease action and can be degraded into fragments. The larger fragments possess polymerase and 3 to 5' exonuclease activity but lack exonuclease activity. This portion of DNA POI1 is called polymerase. Instead of intact POI1, this enzyme is used for polymerase activity as below. See the figure 8. T4 polynucleotide kinase. The T4 polynucleotide kinase catalyses the transfer of 732PO of ATP to 5' terminus of a dephosphorylated DNA, i.e. DNA lacking P04 residue or RNA as below. This enzyme shows both phosphorylation as well as phosphatase activities, therefore, two types of reaction can be used accordingly. In phosphorylation activity, the 7P04 is transferred to the 5' end of dephosphorylated DNA. When ADP is in excess, it causes the T4 polynucleotide kinase to transfer the terminal phosphate attached to a nucleotide from phosphorylated DNA to ADP. By using radiolabeled gamma phosphate, gamma-32PO4, it was found that DNA is rephosphorylated by transfer of gamma-32P from gamma-32P ATP. This shows that the enzyme plays a dual role of phosphorylation activity and phosphatase activity. Due to the presence of both properties, this enzyme is used in radiolabeling of the 5' end of duplex during DNA sequencing. See DNA sequencing method constructing the terminally labeled DNA, phosphorylating the synthetic linkers, oligonucleotide linkers, other DNA fragments lacking terminal 5' phosphates, etc. 9. Terminal transferase. Terminal transferase is also known as deoxynucleotidal transferase. It has been isolated and purified from calf thymus. It has the property to add oligodeoxynucleotide tail to 3,O end of double-stranded DNA fragments. Thus it extends homopolymer tails, and this phenomenon is called homopolymer tailing. For details see Chapter 5. B. Use of linkers and adapters. As discussed earlier, that treatment of DNA with type 2 restriction enzymes results in formation of fragments having sticky ends or blunt ends. Such DNA fragments cannot be ligated with cloning vectors. Therefore, artificially cleavage sites at blunt end can be added as linker or adapter molecules. Chemically synthesized double-stranded oligonucleotides are called linkers. They are added to double-stranded DNA molecules that act as recognition site for restriction enzyme. For example, the following oligonucleotide possess recognition sequence for EcoR1* nucleotides. It can be ligated to blunt end of any double-stranded DNA and cut by EcoR1 to generate sticky ends to DNA fragments, figure 4.5. The chemically synthesized DNA molecules containing pre-formed cohesive ends are called adapters. When recognition site is present within the DNA molecules to be cloned, adapters are used to develop sticky ends. Adapter is used in the form of 5'O so that self-polymerization could not be done, figure 4.5. Thereafter, phosphorylation of foreign DNA ligated adapter is done at 5' end, they join to restriction enzyme treated fragments. See the figure 4.5. Use of linker and adapter molecules in formation of cohesive termini. See foreign DNA, passenger DNA. Foreign passenger DNA is a fragment of DNA molecule which is enzymatically isolated and cloned. The gene is identified on a genome and pulled out from it either before or after cloning. The cloned foreign DNA fragment expresses normally as in parental cell. Thus, the foreign DNA fragments can be procured from a variety of sources depending on the aims and scope of cloning experiments. Identification and characterization of DNA sequences are rather more difficult on its genome than using mRNA, if it is in pure form. If the gene product translated by mRNA is not well characterized, it can be most difficult procedure for cloning. In an average cell or tissue, 1 to 2% of total cytoplasmic RNA population is mRNA, which carry transcripts for coding various proteins. When mRNA is present in low amount, it is rather difficult to isolate cDNA clones. The procedures of isolation and purification of mRNA and getting pure cDNA are given in Chapter 6. Problem statement. End of the chapter 4.